So thanks for being here. You guys know what this is? What is this? What is it?
We're afraid we'll be wrong.
It's a cactus. Is it just one cactus? Are there several cacti there? What's going on?
It's got fascinations.
Yes, it's fascinating fasciation. So this is a crested cactus or a fasciated plant. And you can actually see that some of it is perfectly normal, and other parts of it have these growth, these extra growths, these sort of crowns on top where the tissue kind of branches out in all these different directions. And this is an example of a cancer-like phenomenon in a non-human. It's also quite beautiful, right? The structures that it forms, it's almost like a piece of sculpture. So I've been fascinated by these crested cacti ever since I first encountered them more than 10 or about 10 years ago or so. And I thought, there's something about them that might help us to look at cancer differently, to understand cancer differently. And so this is a picture from a garden that we have at Arizona State University called Endless Forms Most Beautiful. And I'll circle back around to it by the end of the talk and tell you a little bit more about it and how it fits in with the work that we're doing at the Arizona Cancer Evolution Center. But more broadly today, what I want to talk about and the questions that I want to bring up are really about how we can take an understanding of cancer, the understanding of cancer that's really emerging from the work that's being done in this community, and reflect a little bit back on what that can tell us about the nature of multicellular life, and also about the evolution of cooperation itself. Now, we tend to think, oh, well, how can we apply evolutionary biology to understand cancer? And certainly we should do that. But I think there are also some interesting ideas that come from looking specifically at cancer and cheating and cooperation in cancer that can help us understand how cooperation evolves more generally. And then through this sort of feedback between evolution of cooperation, evolution in cancer, I think that will ultimately lead us to a more nuanced approach to prevention and treatment. And I'm going to give you the punchline of the talk today, which is that if we sort of look at the very big picture, we can see that multicellular life evolved to promote and protect cellular cooperation that makes us viable, and limit invasion of cheating from the inside, from the outside, and also potentially to break up cooperation among cheaters. So I'm going to try to pull all of that together today. This is my official title slide, but I don't really like starting with official title slides. It has all my affiliations and all those things that you would expect on a title slide, but we're going to move right along. Okay, so 5 things that I want to talk about today. The first one is that we can understand ourselves as essentially cooperation incarnate, that multicellular bodies are made of cellular cooperation, and it's that cooperation that is really what makes us who we are. Rather than, say, being descended from one germ cell and being made of identical cells, that doesn't really make us who we are. In fact, that's not right. We're not just made of identical cells from the germ cell. So it's that cooperation that really defines us. That in order to suppress cancer, essentially what multicellular bodies have to do is detect and suppress cellular cheating. That if we look at what's happening in cancer cell cooperation and we take what we know from cooperation theory, it's a very, very reasonable hypothesis that what's happening in tumors is probably transient cooperation. I'll talk a little bit more about what the nature of that transient cooperation is likely to be. I'll also talk about how cheating can come from inside, but also from outside. So this connects then to transmissible cancer, but also in terms of thinking about the microbiome and its role in cancer. And finally, I want to make a call really for us all to expand our frame of reference to understand cancer. And those of you who are in this room, you're already doing that. You're taking evolutionary biology, you're taking ecology, you're taking computational approaches and sort of putting them all together to better understand cancer. And I think there are even more opportunities for us to expand our frame to understand cancer better. So let's expand our frame for a moment. Who are we really? What are we made of? We are composed of cooperative multitudes. So we are made of cells that vary, right? We're not made of identical cells. Our cells vary phenotypically because in order for us to be functional multicellular beings with division of labor, our tissues differentiate. And so we have epigenetically different cells, phenotypically different cells. There's, of course, also mutations in our cells, so they're not all genetically identical. We also are chimeras, so all of us have cells from our mothers inside our bodies that got there when our mothers were pregnant with us, and those of you who've had children, you have cells from your offspring still in your body. So we are also made of the cells of our relatives, and we are made of trillions of microbial cells as well that are expressing genes and interacting with our own cells and fundamentally shaping our phenotypes. So when we think of who we are, it's not just cells descended from a germline that are the same. We have to think of all of these dimensions in which the cells that make us up vary. And the thing that really ties it all together is that ideally in a healthy functional state, these cells are cooperating to make us viable. And if we think about what has been the challenge to that cooperative multicellularity, really from the beginning, multicellularity has been facing the potential threat of invasion from the outside, whether it's from microbes or from cellular invaders. So when we think about it and we think of things like transmissible cancer, they might not really be so evolutionarily novel. All right, let me tell you a little bit about my background before we jump into some of the nitty-gritty here. So Carlo mentioned that I come from cooperation theory, and I got involved in cancer biology because I realized that cancer was fundamentally a problem of cellular cheating in multicellular bodies. And so when we look at the problem of how do we support cooperation and how do we limit cheating, there are parallels, really strong parallels between how that works in social organizations, whether we're talking about humans or social insects or bacteria, or if we're talking about cells in our own body and how they cooperate. So as the group size gets larger, the possibilities for the opportunities you can have from cooperation get bigger, as you can divide labor, you can have economies of scale, you can manage risk better when you're larger. But as the groups get larger, the threat from cheating becomes more substantial because any cheaters that arise can exploit the system more readily. And the larger it is, the more there is to exploit. So there's a real parallel between this problem of cheating in cooperative social systems and cooperative cellular systems. So I started my work looking at the evolution of cooperation and movement at the University of Pennsylvania, doing computational modeling of the evolution of cooperation. And then when I was a postdoc at the University of Arizona in Tucson, I started working on cancer evolution. And at some point when I was in Tucson, I must have seen some crested cacti. I don't remember it, but there's quite a few there. So my journey to looking at evolution and cancer has always sort of intersected with these beautiful monsters. Then I went to UCSF, and together with Carlo Mele, we started the Center for Evolution and Cancer there, and we begun these meetings, which Carlo mentioned at the beginning. We're now at the 5th of these meetings. It is now ISEEK as opposed to IBAC, but it's the same group. And I had the opportunity to spend some time in Berlin at the Wissenschaftskolleg with a cancer evolution group that Michael Hochberg convened for a year, where we got to essentially spend the afternoon twice a week just talking about what are the big questions in cancer evolution. And one of the questions that we decided on, so this was back in 2013, I believe, we decided that it was important to look across species, in particular across the branches of multicellularity at cancer, and see what reports there are of cancer. So this was before there really was any work on comparative oncology. So my time at WICO with Michael Hochberg and Hannah Coco and Carlo and Amy Boddy and others was really formative, and you'll see a lot of echoes of that work in the rest of my presentation. And now at Arizona, as part of the Arizona Cancer Evolution Center, we're doing a lot of work together as a team. So during my talk, a lot of the work that I'll be presenting is work that has been done by really the entire team. And I'm also going to weave that together with some work from other labs that's relevant to the bigger picture as well. And then finally, I'll just mention a lot of the ideas that I'm going to talk about today. I've developed further for my book that will be coming out in 2020 from Princeton University Press about looking at cancer as this problem of cellular cheating. So if you're interested in following up more, that'll be available soon. Okay, so back to our big picture, our big question. What can cancer tell us about multicellularity and cooperation? We are cooperation incarnate. If we just look at our somatic cells and we look at what had to happen for multicellularity to evolve, these are really the foundations of multicellular cooperation. This is what happened when we sort of transitioned from unicellularity to multicellularity. There's the evolution of the ability to inhibit cell proliferation, to control cell death, to distribute and allocate resources in the organism, to divide labor, and also to create and maintain sort of extracellular environment that supports the whole system. So these are the foundations of cooperation that are happening on a cellular level that make multicellularity possible. And cells essentially evolved to both cooperate and also to be able to coordinate in this transition to multicellularity. So really the evolution of multicellularity, we can think of it as the evolution of cooperation and very, very complex coordination that requires all this sort of regulation and signaling to make it viable. And what happens with cancer is essentially a breakdown of that cooperation. So as the systems that underlie that cooperation get broken through genetic mutations or through epigenetic influences, the microenvironment, et cetera, what you see is the emergence of these things that we think of as cancer phenotypes, right? Uncontrolled proliferation, inappropriate cell survival, resource monopolization, this sort of upregulated metabolism, dysregulated differentiation, and also the degradation of that multicellular environment. And if we look at work from some other labs, we can actually see that the genes that are responsible for cellular cooperation and for detecting cellular cheating evolved at this transition to metazoa. So as multicellularity was evolving, so was the cellular cooperation and the suppression of cellular cheating. Also, if we look at RNA sequencing data from TCGA, we see that, and this is work from David Good's lab, that in cancer, genes that are associated with sort of more unicellular phenotype are upregulated, and the genes that are associated with the more multicellular phenotype are inactivated. But perhaps even more importantly, you see a loss of coordination between the unicellular and the multicellular genes. So they found, I think it was 12 genes that were kind of at this intersection between the unicellular and multicellular, which suggests that they're playing an important role really in the regulation. And those were the ones that were sort of most out of place, that were most dysfunctional in cancer. So we can really think about cancer in this framework of a sort of disruption to proper multicellular cooperation and coordination. I'm sure you're familiar with the cancer hallmarks framework. So this is a list of the phenotypes of cancer that Hanahan and Weinberg put together. This fits in very, very nicely with this framework of the evolution of multicellular cooperation. So here's a diagram where I've put the hallmarks on the outside and the cooperation and the breakdown of cooperation on the inside. And what we can see is that the breakdown of multicellular cooperation maps on to these cancer hallmarks. And in fact, the dysregulated differentiation is not part of the cancer hallmarks framework, but it's something that pathologists, of course, pay a lot of attention to when they're looking at cancer. So we think that this kind of framework might actually help kind of fill in some gaps in the way that cancer is being sort of talked about in terms of its characteristics across these different fields, from cell biology to pathology. If we look across the tree of life, so I mentioned earlier that along with Amy and Carlo, Michael Hochberg, and others, we did this review of cancer across the tree of life. And we found that cancer was present across all of the branches of complex multicellular life. But there are also a lot of organisms that hadn't been studied yet. And so this was really the beginning and the impetus for a much larger effort that we have now looking at cancer across life. But one thing I did want to mention about this is that when we look at how cancer appears across life, it is always associated with this sort of breakdown of multicellular cooperation. Very often with a breakdown of this sort of division of labor, inappropriate differentiation, and of course with inappropriate cell survival, too much cell growth. All of these things cut across species lines in terms of our understanding of cancer. So this cooperation theory framework offers a much more general way of looking at cancer, one that's not just specific to human cancer or mammalian cancer. All right, so let's talk about cheater detection now. So if we expand our frame for a moment and ask, how do organisms in general solve the problem of cheating in cooperative systems? There are a bunch of classic explanations. So one is that there's some sort of account keeping, reciprocity, where individuals keep track. Another is that individuals who are interacting with kin the genes that code for cooperation can be in the kin. And then there's another explanation, which is that there's some enforcement mechanisms. There's some ways of detecting and suppressing cheating. So through this talk, I'll come back to some of those, but for now, let's focus on the cheater detection in cancer. So in my lab, we work on cheater detection across systems. So this is the work of one of my graduate students, Andres Munoz. He's interested in how do humans detect violation of rules for transferring resources. For example, if you take the benefit, you have to pay the cost. So that's a sort of account-keeping mechanism. But also things like you should only ask for help if you're genuinely in need. And that's a kind of rule that our bodies probably have cells that follow, right? So only signal for blood vessels if there's actually a wound that needs to heal, right? So there's parallels here in terms of the kinds of signaling that can happen in human systems and kind of signaling that can happen in cellular systems. So cheater detection is something that's relevant and necessary for cellular societies as well as human societies. And in particular, when we have larger bodies and longer lifespans, then those are cellular groups where there's greater potential for cheating to arise. And the fact that there's more opportunities for cellular cheating to arise means that there's going to be greater selection for mechanisms that help to suppress that cellular cheating so that the organism can survive long enough to successfully reproduce. And so this brings us to Peto's paradox, which is this idea that cancer susceptibility does not actually scale with body size. If cancer were simply a numbers game, we would expect it to scale with body size, but it doesn't. So what explains why elephants don't get as much cancer, or don't get way more cancer than humans or mice? Well, the evolution of cancer suppression mechanisms, of course. So this is one of the foci of the Arizona Cancer Evolution Center, again, which is a huge team of researchers where we're looking at these questions together. This project is being led by Amy Boddy and also Valerie Harris and Tara are working on, there's many people working on this project together. And what we're doing is gathering together a great deal of data from veterinary pathology databases and others. This is some work that Lisa presented earlier, so I won't spend too much time on it. But in the database that we have, we can actually look at the relationship between lifespan, longevity, and cancer susceptibility, and we're seeing that there is this confirmation of Peto's paradox. So longer lifespan organisms and larger organisms don't seem to get more cancer. So how are bodies detecting the cellular cheating, right? Like it seems like that's what's going on from the comparative data. Well, there are a lot of strategies if we think of it from the perspective of what's going on in terms of the information that's getting processed inside a body. Some of it is going on at the cell level, where cells are monitoring themselves essentially for DNA damage, for misfolded proteins, for abnormal proliferation, for viruses, right? And this sensing is going on through the sort of normal gene expression changes that occur in response to inputs, right? So there's a sort of cellular information processing happening that's allowing for this sort of cell-intrinsic cancer suppression system. But there's also systems that involve the cellular neighborhood. So cells can monitor their attachment to neighbors. They can sense the tissue architecture through sensing the physical forces and other aspects. And they can also sense if cells nearby them are surviving when they shouldn't, because they're processing the information in their environment, both the factors that are around them and the physical forces that are around them. And finally, we have this sort of system-wide surveillance, which includes the immune system and other mechanisms that allow the body to kind of get a sense of if there's any large-scale issues in areas of the body, and then sort of send immune cells or other resources that are needed to those areas. And so some of the ways that these systems can then suppress cancer include like DNA repair and apoptosis at the cell intrinsic level. The neighborhood level, cells can send stop signals. They can stop sending survival signals, that is. They can also send pro-apoptotic signals. And then the system-wide surveillance that includes the immune system actually targeting and eliminating abnormal cells. And that then helps to kind of keep multicellularity from evolving away from this cooperation that helps to promote the multicellular viability. So we can think of genes like TP53 and other cancer suppressor genes in this kind of computational information processing context, where what these cells are doing is essentially taking in information from the cell and from the surroundings and using that to essentially make a decision. And I'm using quotes here just because I'm not saying a decision like we think we make decisions consciously, but I do think they are making decisions in the sense that they're taking in information, they're using algorithms to process that information and then produce some sort of output. So they process information just like a computer might process information. So they're integrating signals from a lot of diverse pathways in order to then decide on the cell fate. So do they complete the cell cycle? Does it require DNA repair? Or is it sort of at the point where all is lost and it should just undergo programmed cell death? So in this way, we can potentially make some sense of a puzzle about TP53. So this is actually something that I've been puzzling on for maybe about a decade, which is if TP53 is so important for cancer suppression, why is it the central node in a network where if you knock it out, the whole network goes down? Right? That kind of doesn't make sense if it's so important for cancer suppression. Why would you make it just this vulnerable node instead of, having it be embedded in a resilient kind of network? So one solution to this possibility, or one solution to this puzzle is that in computational systems where you need to make decisions that are very complex, oftentimes having what's called a bow tie architecture makes the most sense, where you have things coming in all to one central point where they're then processed. And this actually can allow for making a better decision than is possible if the information is going to lots of different nodes. And so for a gene like TP53 and potentially other cancer suppressor genes, making an accurate decision of, this cell is problematic or this cell isn't might be particularly important. And I'll come back to that actually in a few slides. So why is it that accuracy would be so important that we might sort of give up a more robust architecture? So here are some examples of ways that TP53 may potentially be detecting cellular cheating. So we know that TP53 is connected to all of these different sort of sets of genetic pathways that regulate things like metabolism, signaling, apoptosis, proliferation, et cetera. And what may be going on is that TP53 is sort of integrating information to see, is this combination of signals that it's getting sufficient to make it like the cell is problematic? For example, there's a lot of proliferation signals, but there's no sign that there's actually a wound that's being healed. Well, then maybe there's a problem. But if there's a lot of proliferation signals and there's also signs that there's a wound there, maybe it's okay. So let's come back now to this question of what's happening with genes like TP53 in terms of suppressing cancer. So some work that was done by Lisa Beglin and Carlo and many others who are here today has shown that elephants actually have multiple copies of TP53. So they have potentially a more robust system than we as humans do. And elephant cells are actually more likely to undergo apoptosis in response to radiation than human cells. So it suggests that this is a cancer suppression mechanism. And if we look at the evolution of body size in elephants by comparing with woolly mammoth and mastodon genomes and others, we see that the copy number of TP53 actually increased as body size increased, which suggests that selection on TP53 might have been driven by that increasing body size. And there's also some work that was just published a couple of weeks ago by Mark Tolis and some others on our ACE team, showing that there has been positive selection on cancer-related genes in giant cetaceans, so in humpback whales, for example. There's accelerated evolution of the apoptotic pathway, positive selection on genes that have to do with the cell cycle checkpoints, cell signaling, and also cell proliferation. So having mechanisms for suppressing and detecting cheating cells seems to be something that large, long-lived multicellular bodies need to do, but not always through the same mechanism. So there wasn't any evidence in the whales, for example, of enhanced TP53. So there's another side to this, though. And remember, I sort of referred to the need for accuracy in detecting cellular cheating and sort of activating TP53, for example. And that's because detecting and responding to cellular cheating can have costs as well. If an organism is sort of too ready to apoptose cells that might be cancerous, then a lot of cells that might have been perfectly healthy go to waste. You might also have issues with too much inflammation if these mechanisms are sort of too likely to get activated. And there can also be trade-offs with things like development and growth and tissue maintenance if these cancer suppression systems are too easily activated. So we can actually take this into a signal detection framework where we consider what are the costs of a hit or a miss. So you could sort of think of this as TP53 making the decision, is this carcinogenic, yes or no? And if TP53 basically makes the decision yes, it looks like the cell is carcinogenic, then if the cell, if it does not actually respond and there's a miss and the cell survives, then you can get an increase in cancer risk, right? Because that cancer cell was not actually killed. On the other hand, if TP53 activates and shuts down the cell, apoptosis, that prevents cancer. So, and we tend to think just of, well, is TP53 or other cancer suppression mechanism, are they catching cancer? Yes or no? And that's the relevant thing. But there's another side to it also, which is you can have cells that aren't carcinogenic, and if they're sort of correctly not apoptosed, then that avoids unnecessary costs. But if they are apoptosed when they would have not caused cancer, then that can generate various unnecessary costs. And so we can actually use all of the tools of signal detection theory to try to get at these questions. And another way of thinking about this is just in terms of trade-offs and trying to balance various potential outcomes. So one way of thinking about what an organism is trying to do as it sort of traverses this tightrope of life is balancing the need to grow and reproduce and replace cells that are broken with the need to shut down cells that are doing things that they shouldn't do. And you can almost think of this as sort of balancing this tightrope between sort of chaos on one hand, chaotic, cancerous pit of destruction, and then on the other hand a sort of stagnant, freezing area where you can't actually do anything, right? If cells are if shutting things down all the time, then you can't go anywhere. One example from some modeling that I did with Hannah Coco, Amy Boddy, and others, we essentially found that if we vary mortality and the amount of intersexual competition, that you can get selection for lower cancer defenses. So in other words, if organisms are competing with each other very intensely for mating opportunities, then that can actually select for lower cancer defenses because greater investment in mating opportunities and fertility can have these trade-offs. So all this is to say that there are a lot of important open questions about what the computational architecture is of detecting and responding to cellular cheating in multicellularity. There's a lot of tools that we can use from cooperation theory, from signal detection theory, from evolutionary biology more generally, and I think there are a lot of really important opportunities for sort of taking this strategic approach to thinking about, what's going on in terms of the evolution of cheating and cooperation, and also taking a very sort of information theory kind of approach to understanding what is actually being computed in these genetic networks. And I think this is one of the most exciting areas for future work. So let's talk about transient cooperation in cancer for a few minutes. So one of the big puzzles in evolutionary biology is, of course, this question of how can cooperation evolve and be maintained when cheaters can get the evolutionary upper hand, right? If cheaters do better than cooperators in a group, then cheaters should always take over. So how does cooperation evolve in that kind of world? Well, there are a bunch of different ways that cooperation can evolve. Traditionally, I talked about kin selection and reciprocity. Those are solutions that have been talked about a lot. Also, this idea of sort of detecting cheating or choosing partners. But I also want to mention positive assortment. But I'm going to give you kind of a 1-minute cooperation theory 101. So when we look at these different explanations for the evolution of cooperation, they all sort of come down to this question of, what's the likelihood that individuals will sort of share the basis for that cooperation. In the case of genetic relatedness, it's sort of looking at the likelihood that they will share the genes that code for cooperation. When we look at reciprocity and division of labor, what we're looking at is essentially the likelihood of future reciprocation, multiplying that by the future benefit from that reciprocation. When we're looking at partner choice and cheater detection, essentially what it comes down to is, what's the likelihood of interacting with a cooperator and the benefit of interacting with a cooperator. And what all of these have in common, actually, is that it's about how likely is it that a cooperator is going to interact with another cooperator more likely than would be expected by chance. And if that's the case, then you can get the evolution of cooperation. So even though it seems like all of these explanations for the evolution of cooperation are based on very different mechanisms, it all comes down to this very same logic of if cooperators are more likely to interact with other cooperators, then that can actually sort of provide the scaffolding that allows cooperation to evolve. Another way to think about this same idea is that cooperation can evolve any time you have two or more cells that positively influence each other's fitness. So my colleagues and I put together this framework that's a very general framework for thinking about the evolution of cooperation based on what we call fitness interdependence. This is an idea that's actually been around for several decades in different literatures in evolutionary biology and social psychology and even sociology. And so we've tried to sort of pull it all together. And essentially what it comes down to is that individuals can influence each other's fitness. And to the extent that two or more individuals have positive effects on each other's replication of their genes, then you can get cooperation. So I want you to imagine here that you're on this Ferris wheel and it's going around and around, and you're up there in number 12 with somebody. So when they rise, you rise. When you go down, they go down. You're exactly positively fitness interdependent with each other. On the other hand, if you're in car 9 and your counterpart is in car 3, when you do better, they do worse, right? So that's a zero-sum kind of situation. So we can actually take lots of different kinds of scenarios where individuals are sort of tied together in their fitness interests and represented in this general kind of framework. And this applies to humans interacting just as much as it applies to cells interacting in a tumor or in a petri dish or wherever. So when we think about cancer cell cooperation, we have to look at it in this context of multicellularity, right? So they have this whole toolbox of multicellular cooperation they can draw on. And one of the things that's probably happening is that they're using these tools to facilitate a sort of more collective cellular cheating strategy. But they're going to be using the same general mechanisms that happen in other systems, right? There's not something fundamentally different about cooperation among cancer cells. So we know that one of the ways that cancer cells cooperate is by producing diffusible factors like growth and survival factors. They can also protect each other from the immune system. They can recruit blood vessels that feed not just themselves, but also neighboring cells. And they can also hijack the immune system to provide benefits for neighboring cells as well. Now, one thing that's really important to sort of connect with all of these ideas is that oftentimes where you see cooperation is at these frontiers, invasion and metastasis. So we know that cancer cells actually cooperate and they coordinate their electrical signaling to invade tissues. We've also seen that clusters of cancer cells are better at metastasizing than single cells. And if we look at this in the broader context of evolutionary biology, we see that in general, selection on frontiers in harsh environments can oftentimes favor cooperation. So cooperation among cancer cells may be selected for in particular at these frontiers where invasion and metastasis are going on. I'm going to actually just jump ahead a little bit because I don't have that much more time left. So one possibility of what might be going on in cancer is that you can actually get selection favoring cheating sort of in cellular groups when those are relatively stable. But then cells can evolve to move, and I have some modeling results that show, yes, cancer cells evolve to move in response to low resources in the depleted environments that they themselves create. They also evolve to conditionally move more readily in these models. So you can have cell motility and invasion happening because the environment's sort of gotten depleted as a result of the cheating. Then cells, again, are having these challenges of surviving at these ecological frontiers, which then may favor cooperation, right? And then once you have those clusters where cooperation is happening, cheating can start to arise again. So it's possible that there is this sort of dynamic kind of churning happening where cooperation and cheating are kind of getting selected in turn. This is a hypothesis for what might be going on. I think it's something that we should be considering as we are starting to get better tools for looking at the evolutionary dynamics during metastasis. All right. There are a lot of strategies that we can potentially use for cancer treatment that come from this approach, and one of them is actually trying to target the cooperation among cancer cells and break that up. For example, by trying to break up the adhesion, trying to interfere with the signaling that may be allowing them to coordinate. All right. Cheating can come from within or without. So we've already talked about how cellular cooperation is not just the somatic cells of our body, right? There's a lot of other genetic individuals in there. Well, cheating also can come from outside. And one thing I think is very important to cancer but has not really been touched on at this meeting is the role of the microbiome. So microbes are actually, there's evidence that they are cooperating with cancer cells and that this cooperation is relevant to progression. So, for example, salmonella can produce factors that lead to base pair shifts and tumor proliferation. E. coli produces hydrogen sulfide, which can reduce mucus production, break down the intestinal barrier, directly cause DNA mutation. And fusobacteria have been shown to be associated with increased inflammation and leaky junctions. Now, the ways that cancer cells can affect, the way that microbes can affect cancer cells, some of those are quite direct, like initiating cancer through damaging DNA, initiating cancer by sort of increasing the amount of cell proliferation, providing growth factors. So cancer cells and microbes will provide growth factors for each other. And cancer cells and microbes can also protect each other from the immune system. There's also indirect ways. So by altering the intestinal barrier and the biofilms in the intestine, microbes can indirectly increase cancer risk. They also can induce the proliferation of host cells, which can expand the microbe's ecological niches. So for example, if they're increasing cell proliferation in colonic crypts, right, those can extend the colonic crypts and create more of an environment for the microbes as well. Microbes can prompt a shift to more metastatic phenotype through EMT, for example. They also produce quorum-sensing molecules that can contribute to metastasis, and they can also alter epigenetics. And there's a positive side to this too, though, don't worry. Some microbes have been actually associated with protection against cancer. So the short-chain fatty acids that bifidobacteria produce actually help to maintain the barrier, and they help keep those junctions tight. Lactobacillus produces vitamins that actually help to properly methylate DNA. And Saccharomyces produces compounds that reduce DNA damage and inflammation and can inhibit tumor growth. So we can think about potentially some of these microbes as cooperating with the normal cells of our body to better suppress cancer, while other microbes may be cooperating more with the cancer cells in order to promote their mutual fitnesses. So if we think about the evolution of multicellularity, we can think about cellular cheating more generally. So I brought up this issue that transmissible cancer is an ancient problem. So in the transition from unicellular life to multicellular life, there is this phenomenon that some cells could benefit from actually parasitizing other groups of cells, so very early on in the evolution of multicellularity. So germline parasitism, this is where cells in sort of very early multicellular life might invade another multicellular body and become the germline, or stem cell parasitism, where cells might invade and find their sort of place in a stem cell niche and then sort of use that to proliferate. So these may be very early versions of transmissible cancer. We know that transmissible cancers aren't so uncommon today, and many of the talks that we'll see in the rest of the session will give you a much better idea of that than I possibly could. And Mel Greaves' lecture yesterday, he talked about just how rare transmissible cancers are in humans, but they're not absent. And one of the things I think that's important to think about is, well, when we do see contagious cancer, when does it happen? And it happens when the immune system is somehow challenged. So you can have a break of barrier, like the skin. You can have suppression of the immune system, or in some cases, hijacking of it. And when you have hosts that are more genetically homogeneous, it can be harder for the body to essentially detect, for the immune system to detect. So contagious cancer, transmissible cancer, when we see it, it seems to be associated with this sort of breakdown of our immune detection. And so it's possible that some of the aspects of immune surveillance that we think of as cancer suppression mechanisms may have some very, very deep evolutionary roots in also protecting against transmissible cancer, at least originally. So I think these are all important questions that we should be thinking about. So multicellular life has actually co-evolved with cancer, including transmissible cancer, since the beginning. And that should be part of our frame when we're thinking about cancer more generally. So we might not want to just take for granted that, transmissible cancer never happens in humans. Well, yes, it's extremely rare, but that might be because we have mechanisms that help keep that from happening. There's some interesting work suggesting that, perhaps the evolution of sex might have arisen in order to help protect from transmissible cancer. This is work by Fred Thomas and others who are here today. And the idea is that sex could have reduced the transmission of malignant cells to offspring. Now, I want to bring up microchimerism again, because this is another potential situation where you have transmission of cells to offspring. This is work that I've done with Amy Body and others, where we looked at microchimerism in pregnancy. And so we know that when a mother is pregnant, there's a transfer of cells from mother to fetus and vice versa. And we can ask, does microchimerism, is there any evidence that it contributes to cancer? So what Amy and I found, we did a thorough review of the literature on microchimerism and maternal health, and I'm just highlighting a few pieces of this here. What we found is that microchimeric cells sometimes appear to be contributing to cancer, associated with higher cancer risks, and other times they seem to be protective against cancer. They're associated with lower cancer risks. So these items here in the box show just some examples where there was an association either with less cancer or with more cancer in the breast and thyroid, and also in the lungs and in reproductive tissues and in the colon. So how are microchimeric cells contributing to cancer? We don't know for sure, right? Because the data sometimes show a positive association, sometimes a negative association. But it may tie in with some other issues like James Gregori's talk, where he talked about differences in sort of cell competition inside the tissues as we age. So perhaps the age of pregnancy is playing some role. Maybe sometimes microchimeric cells are actually able to take up ecological space that might otherwise be occupied by cancer cells. And maybe other times microchimeric cells are actually generating situations where they're changing the microenvironment in ways that increase cancer. And maybe sometimes they're actually the agent of cancer themselves. So we don't have good enough data now to know whether that is the case or not. So there's open questions there. So just a few last words that I think if we're going to really understand cancer, we have to continue to expand our frame of reference. And that doesn't mean that we always are just expanding. Sometimes we also have to focus down and ask very particular questions. But I think we want to continue to expand our frame of reference. So, all of us here, we think about things like, how does cancer look across the tree of life? We think of how, cancer has emerged in the history of life, and then also, thinking about the sort of diversity of us as organisms and what makes us up. But we have to also continue to expand our disciplinary engagement. So, we, of course, use a lot of evolutionary theory. Ecology, over the last few years, has been making a big difference in how we understand what's going on in the tumor microenvironment. But I think there's also even more opportunities for us to expand and start thinking about other areas like, for example, the arts and illustration, and how can we actually represent some of the things that we're doing in visual ways that can then feed back and help us to understand what we're doing better. So for us in ACE, in the Arizona Cancer Evolution Center, arts and outreach and education are an important part of what we do. So this is a picture from our cancer garden. Carlo Mele's leading a project actually representing cancer in music. And Carlo mentioned my podcast, Zombified. We're also doing a podcast that's being led by Pam Winfrey about cancer, which is a combination of a sort of narrative about someone dealing with cancer and scientific questions and engagement with scientists about cancer. So I want to just show you a few photos from our garden. So this is the plan for the whole garden. That little black spot there is a person, so you can see it's quite large. And these are some pictures from the installation and the planning. It was an amazing effort that included not just members of ACE, but many people who are part of ASU landscaping and grounds, and many of them have been affected by cancer themselves. And so when you do a project like this, you really see how important the issue of cancer is in many people's lives. And for us, one of the important things about this garden is not just that it sort of represents cancer in something that's not human, right? And that kind of helps us to think about and start talking about cancer in a way that's maybe a little bit less threatening. But it also underlies this capacity that life has to coexist with cancer. And I think that opens up the possibility for new conversations about things like adaptive therapy, where what we're doing is trying to control the tumor and support the patient, as opposed to just trying to eradicate the tumor. And so I just want to end with this quote from Darwin's Origin of Species, where he says, "There is grandeur in this view of life, from so simple a beginning, endless forms, most beautiful and wonderful, have been and are being evolved." And this was the inspiration for the name of our garden, which is Endless Forms, Most Beautiful. Thank you to all of you, to the entire ACE team, especially the people who've contributed to all of the work that we've done on comparative oncology and on really bringing cancer and the arts together as we're trying to do now. And I'll just make a little plug that the next iSeek meeting will be on the art of cancer. So we will be exploring these ideas more through talks from people who you might not expect at a cancer meeting necessarily. So thank you all, and I think I have time for maybe one or two questions. Yeah, thank you.
Thank you, Athena. The question is how environmental harshness can break down the group division or the cooperation?
Yeah, so the question is about when does harshness break down cooperation? So this is a question that we've actually looked at in my work with humans, so I don't know to what extent it's been looked at in cellular populations. But essentially, what looks like happens is that in harsh environments where there's risks, individuals that are able to sort of share risk, so essentially, if you sort of make an agreement to provide social insurance for each other, then that makes it more likely that everyone survives. And in fact, individuals who participate in those systems do better individually as well, because they're participating in this sort of insurance system. Now, challenges happen when that harshness is persistent over such a long period of time that everyone kind of goes below a level where they could sustain themselves. And so it's really in that sort of very, very slow burn where there's no way out and the harshness is just persisting that you can get a breakdown of a cooperation. But in situations where it's not that persistent, it's temporary, then cooperation can be very much favored. Yeah.
What about the mortality rates in non-human species because of cancer?
Can you tell me a little bit more, the mortality?
Yes, I mean, cancer can coexist with non-cancer cells in non-human species, right?
Mm-hmm.
Is there any intrinsic difference in the mortality that produces cancer?
Yeah, so if we look at species like plants that have fasciation, they are affected by the fasciation. So it does oftentimes affect their flowering, so they don't reproduce as readily. They're usually more delicate and they require more care. So I think that's actually a very strong parallel with cancer in humans too, right? That it can have these effects and people, require more care than if they didn't. But I think there's a lot of very promising work now on these evolutionarily informed approaches like adaptive therapy, which suggests that, yes, it is possible to actually extend life for a long time by focusing on control of the tumor rather than on the treatment or on the eradication of the tumor.
I think that I can't help to think all the time is making analogies between multicellular organisms and societies and things like that.
Mm-hmm.
Do you have anything to say about parallels of cancer in societies?
So sort of about taking the analogy back to human societies. Yeah, so that's a challenge, right? Because we, as humans, we don't function as a multicellular body, right? I mean, yes, there's cooperation, and we do things like share resources, and allocate them based on, sometimes based on need, depending on your political leaning. But I think it's hard to sort of bring the analogy of cancer fully back to human societies just because we haven't evolved to cooperate on the level that multicellular bodies have evolved to cooperate on. But at the same time, that doesn't mean that it's completely worthless to consider, well, maybe should we be taking better care of our environment like cells in a multicellular body do? Should we be considering how we're functioning together to lead to longer lived groups that are viable? I mean, I think all of these things, there's some leads into thinking about human cooperation, but I think we need to be cautious with sort of pulling the analogy back in full force.
So, do you think that cancer is, in a way, like a necessary condition of cooperation, as a sort of like some sort of like way of balancing or, error managing or something like that?
Yeah, so I think that the emergence of cheating in large-scale cooperative systems, in some ways we can say that's an inevitability, that it will arise. I don't think it's necessary. I don't think it provides something that's functional for the cooperative group. I think it's just something that happens when you have large-scale cooperation and you don't have mechanisms in place to control that cheating. When there's an advantage to cheating, then it's going to expand in the population. It's just sort of simple evolutionary biology. And yeah, I think it's something that multicellular bodies have evolved to deal with, so we have a lot of mechanisms to keep it under control. But I don't think it serves a function for our bodies that it's there. Yeah. Thank you.